previous video, I just laid out some fundamentals on answering questions on protein sequencing via endopeptidase digestion. In this video, I'm actually going to discuss this harder problem that I haven't really seen dissected in a YouTube video before. So that's what I'm doing right now. So in this question, it's actually very similar to the one we just did earlier. So just to refresh you, this is what we did earlier. But you'd notice that I have here elastase, so it's like a different endopeptidase. So we're just going to briefly look at what it does, but it's actually very similar to what we've done earlier in that it cleaves. Okay, this one cleaves A, G, S, and V. So adenine, glycine, serine, and valine at the C terminus. So it's still at the C terminus. Okay, trypsin and pepsin, we've seen that before, so it's the same thing. It cleaves a particular amino acid. In this case, it's arginine and lysine at the C terminus also. Pepsin cleaves leucine, phenylalanine, tyrosine, and tryptophan at the N terminus. Right? So this is at the N terminus. We're just going to briefly take a look at what that means again. So when we mentioned that this is on the C terminus, what we meant by that is, for instance, we have alanine over here. So let's say you have alanine there. Then we have elastase working on the peptide. So it's going to cleave off at the C terminus. If, for example, we have pepsin and we have phenylalanine F, cleavage at the N terminus means that we're cleaving off somewhere here. Okay, so that's the fundamentals of that. So now we're going to take a look at this problem. Notice that for elastase we have an alanine here, and we also have a glycine over here. For trypsin, we have R and K again, so arginine and lysine. For pepsin, we have phenylalanine, right? So phenylalanine and tyrosine. Okay, there's a problem here, because if you were just looking at this casually and like you saw, okay, so there's two cleavage points. If you have a straight line, right, and you have A here or G here, right, you'd imagine that when you cleave those different side chains, you're going to get three parts, right? This is 1, 2, and 3. Same thing with trypsin, right? So when you cleave at lysine or arginine, like let's say hypothetically this is K, you cleave there, and you have arginine here, you're also going to get three parts, right? So this is 1, this is another part, 2, and then you have another part here, 3. So this one doesn't necessarily make sense. When you see this problem at first, you're thinking, okay, there's something wrong with the question, but actually there isn't anything wrong. It's just that you haven't seen a question quite like this, in that you don't have a linear peptide, you have a circular peptide. So I first encountered this in an exam before, and it was like super crazy for me because I was just trying to answer as much as I can, and this gave me such a headache since I wasn't given an example where the peptide was circular before. But it's actually quite possible, where you have the N terminus and the C terminus reacting with one another, and you're going to get a circular peptide. So this is actually what you have here. So once you understand that you have a circular peptide, you're more or less okay, right? So that's half the battle done. The problem now is, how do you work with circular peptides? So this is working with protein sequencing via endopeptidase digestion of a circular peptide. Okay, so let's start. So for elastase, as you can see, you form here a tetrapeptide and a pentapeptide. So there's still 9 amino acids here, like the previous video. But this time, it's circular. If it's circular, that means there isn't really a start and an end. It's a circle. But we know that the N terminus is in the left part of the amino acid and the C terminus is in the right part. Okay? So here, for instance, we have AFC RE KGYM. So this would imply that G, right, should be a cleavage point over here. Okay, so it will cleave at the C terminus. So whatever amino acids you have, so we have K, Y, and M, right? And then it's going to be here, it's going to go on a circle. Being on a circle, right, that would necessarily mean that your A, right, your alanine should be cleaved off somewhere over here, right? So for you to form that tetrapeptide, right? So alanine has to be cleaved off over here because this is the C terminus of alanine. This means that the rest of the amino acids can be placed, but we're sure that glycine, G, and alanine, A, is on that position, right? Or are on that position. So we can just put here the other amino acids that we are not sure of yet, or just put them there, CRE. Okay, so that's the first part for elastase. Okay, we know where G and A are. Now let's take a look at trypsin. For trypsin, okay, we have ACMYK, EFRG, so R and K. So we see them here. So we have R and we have K and we have R over here. So now we know that we want to get EFRG. So that's this one, R, F, E, and G. Okay, but it cleaves off at the C terminus, so that means it will cleave off somewhere here. Therefore, okay, that would mean that C should be here and R should be here. So you can have F, E, G, and then something should be cleaved here, and that something would be K, right? So now we know where those different amino acids should be. So let's write that down. So we know G is already here, and we know A should already be there. Okay. The C, cysteine, can only be here, right? Because as we've seen earlier, R would be cleaved off in the C terminus. So your cysteine can only be placed here. It should contain your E, F, and G. So this would mean I have E and F, and I would also have K here, so I can cleave off, forming my tetrapeptide over here. And this is my tetrapeptide. I would form also a pentapeptide here. So that would mean that M and Y should be over here. Okay. So if we've already established that, okay, so originally G and A are there, we know that R should be here and K should be there. C was sandwiched between the A and R, so we know that this is the only position it can take. So what we are left with are methionine, okay, methionine, tyrosine, glutamic acid, and phenylalanine. So those are the 4 different amino acids that we don't know where to put. Notice that this is clockwise direction. So now we already know those 5 different amino acids. Let's take a look at pepsin to finalize the positions of M, Y, and E, F. Okay, so let's write down what we already know. So we know K, G. We also know A, C, R. Okay, now for pepsin, pepsin is going to cleave at F and Y. So this is somewhere here and somewhere here. Notice that I'm going to produce GKEY. So GKEY, this is the the cleavage of Y, right? Because this is cleaved by pepsin at the N terminus. So I need to cleave it off somewhere here. You have Y, you have K, you have G, and then you have E. So this would mean that E and F should be interchanged, right? So we have here Y. So there's a cleavage site over here at the N terminus of Y, YKG, and then we have E. Right? The F would be cleaved off at the N terminus, right? So we have FRCA and M, because that's the only place where you can have the methionine over. So now, okay, so let's see, we determined KG, AC are before. We know that Y should be over here so that you can form this tetrapeptide, right, as such. And F should be here so you can form this other peptide over here, right? This pentapeptide. Therefore, M should only be here. So now we've accounted for all of the different amino acid residues in this nonapeptide. And in fact, this is a circular peptide. So this is a circular peptide. Okay, so they're all linked together as such. So the crux of this problem is really understanding that you're dealing with a circular peptide. Because if you don't know that you're really dealing with a circular peptide, it's going to be weird. But your biggest clue here is that you have one less segment or fragment that's formed based on cleavage. As we've mentioned earlier, we have elastase over here, we have two cleavage sites. If you have two cleavage sites, that would actually tell you that you should have three different fragments, right? It's an endopeptidase, after all. But you're only seeing two fragments. You see that for trypsin, you see that for pepsin. So, therefore, what you're dealing with is a circular peptide. And I haven't really seen a problem that tackles this before, but this is something that could actually be asked in an exam. So, thank you very much. I hope you learned something about protein sequencing via endopeptidase digestion here. So, thank you very much for watching, guys.